Welcome, everybody, to the last seminar of the semester. Yes, we're almost at the end. Yay! It's my pleasure to introduce our seminar speaker today, Dr. Liz Alter. And then just to tell you a little bit about Liz and her background. So Liz is a marine evolutionary biologist. She's a professor currently at CSU Monterey Bay. And she also has a joint appointment as a research associate at the American Museum of Natural History in New York. Right, for her educational background, she got her BA from Yale University, which I didn't realize until I threw your CV, but we were Ivy League rivals at the same time, but I guess we can still be friends now. She then got a Master's of Environmental Science, Policy, and Management from UC Berkeley, and then was in Monterey for a long period of time, where she did her PhD at Stanford at the Hopkins Marine Station, working mainly with Steve Palumbi. After that, she did a postdoc at the Natural Resources Defense Council before coming to CSUMB. She was a professor for 10 years at York College and the City University of New York. Right? Liz has an extensive background mentoring graduate, undergraduate, and K-12 students in a variety of programs. Right? And she's going to be taking her first master's students into the Moss Landing program in the fall, and it was one, two, I forget. One, yes. And I will be the co-advisor for that student, yes. So that's going to be really exciting. So Liz, she's also really excited to get involved, to get to know you all, because she's going to be involved in mentoring students in our program going forward. So that was kind of the impetus for me to get her to come and give a talk today so you get to know her. Right, Liz's research focuses on understanding how biodiversity is generated and maintained. Basically, she's doing evolution, right, and mainly using the tools of genetics and genomics to do so. And she's worked on a variety of aquatic ecosystems, from oceans to estuaries to rivers, and on a broad diversity of taxa. I was looking through her CV. She's published on a variety of microbes, evolution of COVID, protists, lichens, plants, fish, which is mainly what she'll talk about today, sea turtles, and whales. She's published from viruses to whales, which is crazy. And I could, yeah. And I was thinking, is she trying to get the EGOT for working on the most kingdoms of life? And so we need a new acronym for that, but I think you'll win it. Right? And then basically for her PhD work, again, Liz did a bunch of pioneering work using molecular tools to reconstruct the historic population sizes of whales before whaling, using DNA forensics to identify the actual fish species sold as red snapper. Hint, it wasn't ever a red snapper, it was always something else. Right, she's done a lot of interesting work on adaptive radiation of cichlids in Africa, which I think we'll hear about some today, the effects of climate change and urbanization on marine systems, and the utility of eDNA to study fish diversity and conservation. Right, Liz currently has an NSF Career Award to study the genomics of rockfish undergoing rapid speciation, which is some of the work she's pursuing now. And then on top of all that, right, she's also a commissioner on the Fish and Wildlife Advisory Commission of Santa Cruz County, as well as the Scientific Advisory Board of the Billion Oyster Project. That is a lot of oysters, right? Which seeks to restore coastal ecosystems, right, while training high school students how to do marine science. So with that, really excited to have Liz talk, and she's going to tell us about genomic investigations into the evolution of hyperdiverse fish clades.
Great. Well, thank you so much to Scott for the invitation here, and to all of you for coming on a Thursday afternoon. I'm happy to present some of the work that I've been doing for the past 10 years or so, focused around the evolution of hyperdiverse fish clades. My goal in our time together is to explore some of the features that may drive diversification in two clades of fish, cichlids and rockfishes. So this is the first time I'm trying to kind of do a relatively fast talk, bringing all these themes together. So we'll see how it goes. But I'm hoping to also impart how using genomic tools can really help us better understand these very speciose groups. And in the process, I'm going to present some of my past work, some of my ongoing work, and then try to synthesize some tiny fraction of the amazing wealth of data that's out there on both of these clades. All right, so before diving in, I just wanted, since I'm relatively new here and would love to, like Scott said, love to get more involved in the Moss Landing community and hope to meet many of you and collaborate with many of you. Just wanted to give a little more of an overview of what we do in our lab. So today I'll be mainly talking about the first kind of theme on this slide, which is the evolution of new species. And so we work on that in Congolese fishes and cichlids, and then in rockfishes as well. But we also do some other projects. So we use DNA from both modern and ancient specimens of marine mammals to try to understand how things like climate change have affected populations in the past. And then finally, a big set of projects in my lab used environmental DNA to look at patterns in biodiversity. So for those of you interested in any of those, would love to chat with you more at some point. Okay, but ultimately, primary motivation in the research in my lab focuses around this question. Why is the diversity of species distributed unequally across the tree of life? So for generations of biologists, since Darwin and since Wallace introduced the theory of evolution by natural selection, questions have remained about why it is that some clades are so rich in species while others are relatively depauperate. Some have diversified explosively, and we'll see cichlids are kind of a prime example of that, while others have remained kind of phenotypically unvaried for millions of years. So despite the century plus of study that's happened since Darwin, we still have a lot of questions about the causal factors behind these differences in diversification rates. We have a reasonable understanding of how new species originate, but I think we can all agree that given the biodiversity crisis that's facing our planet, we need a deeper understanding of the factors that determine speciation rates and species richness. So to understand organismal diversification, we need to be able to unravel how genomic level variation is connected with phenotypic evolution. And that requires a comprehensive understanding of how organisms evolve and function and interact with other organisms in their environment. Adaptive radiations, so these are radiations that are characterized by explosive diversification, usually ecological, morphological diversity resulting from adaptation to a new or sometimes previously underutilized niche, really have a unique advantage in helping us to understand the study of biodiversity and how it evolves. So a couple iconic examples here include Darwin's finches, which Scott is intimately familiar with from evolution course as an undergrad, and anoa lizards in the Caribbean, honey creepers in Hawaii, and of course cichlid fishes in East Africa. So these adaptive radiations combine the benefits of both lab and natural selection, natural model species for investigating how organisms diversify. And we can add rockfishes to this list, although that's not without a little bit of controversy whether they count as an adaptive radiation, but I'll make the argument that they fit into this group well. So what are some outstanding questions in speciation research? First of all, we still have relatively little understanding of how new species form when gene flow hasn't stopped, right? So we define speciation as the creation of new lineages that are distinct from others, and that results from reproductive isolation. So what happens when you don't have reproductive isolation? You have this process that's ongoing, but gene flow has not ceased. So the model has changed since in the last couple of decades, whereas there used to be a quite heavy focus on allopatric speciation, the creation of species in the absence of gene flow. Now we understand that speciation is possible and even common, perhaps, in the presence of some level of gene flow between diverging lineages. And ecological speciation and gene flow seem to be very prevalent, actually, in adaptive radiations, like the kind we'll be talking about today. Second, how might ancestral variation play a part in adaptation? So ancestral allelic variation, if it's retained, can also play a big role in evolutionary potential. As we obtain more and more genome-wide data from non-model lineages, we can see that many adaptive radiations are not necessarily driven by de novo mutations that are occurring, but rather by a deep pool of standing variation in those species. And then third, what do changes in the genome look like as radiations proceed? So there's now a pretty hefty body of theoretical modeling that predicts that rapid diversification may be accompanied by so-called islands of speciation in the genome. That is, regions of the genome that are highly differentiated, even between very closely related species that are still sharing genes. And those models also predict that the number of underlying loci that are represented by those islands will be relatively small. So we'll come back to that point later. And then fourth, what might be some intrinsic features of genomes that could influence speciation? So in many cases, we see there's environmental drivers of radiations, and there's particular features like, in the Galapagos Islands or Hawaii, the lakes of Africa, that may help drive isolation of lineages. or drive adaptation to particular niches. But not all clades are diversifying in these environments in the same way. So, for example, the Great Lakes of Africa have plenty of other fish lineages besides cichlids that are nowhere near as diverse. Right? So what is it about cichlid genomes that is special? Are there features, mutation rate, gene duplications, et cetera, that might give us some insights into why certain clades speciate so fast? So examining that, what's called the genomic substrate for diversification, is now a major kind of focus of speciation studies. Okay, so with those questions in mind, we can leave behind the rest of the vertebrate tree, and we'll start to tighten our focus in on some of the hyperdiverse clades within the ray-finned fishes. So acanthomorphs are the most diverse clade of vertebrates with over 18,000 species. And this clade includes some of the most well-known examples of adaptive radiation, including our favorites, the cichlids and rockfishes, as well as others like Antarctic notothenioids and a bunch of fun catfishes and other things. This is a time-calibrated fish tree of life where you can see these little, I don't know if I have a pointer. I can just hop up here. So here we have Ovolenteria eupercaria representing clades with, that are extraordinarily diverse, shown by that yellow color there. And more than just the rate of diversification, as shown in this study from Roboski et al. On clade-specific evolutionary rates in fishes, cichlids and rockfishes turn out to be among the top 10% fastest of fishes for both the rate of speciation and the rate of phenotypic evolution, here using size evolution as a proxy. So what you can see here is that terminal branch lengths are proportional to speciation rates, and tip size and colors represent extant species richness and the rate of body size evolution. So you can see that both cichlids and our rockfish here are right up there with the best of them in terms of the rate of both diversification and phenotypic evolution. So we can establish a few baseline facts and comparisons to begin with. Think about what these two clades have in common and what might be different. First, we have to acknowledge that cichlids are absolutely unparalleled when it comes to diversity. They're the OG of diversity in every way. They're the most speciose vertebrate family, so nothing else really comes close. Nevertheless, rockfish are nothing to sneeze at, given that they are a single genus and a marine clade, so with lots of species living in sympatry. So with that context, rockfish are, have very impressive diversity. So cichlids, obviously freshwater and a center of diversity in East African lakes, whereas Sebastes, the genus, is marine with a center of diversity in the Northeast Pacific. They have in common that there are quite a lot of species pairs or ecomorphs that are depth separated. So that's quite an interesting feature. Now the scale of depth obviously is quite different between ocean and lake. Nevertheless, we do see some common adaptations to depth in terms of their molecular evolution, including the evolution of things like opsins, eye-related genes. To put cichlid radiations into the context of our own species, so cichlids have been diversifying with a pulse in the last 5 to 7 million years. So approximately 2,000 species of cichlids have evolved in that time, and that's about the same time that the human-chimpanzee split occurred. So 2,000 species in that time of our own kind of hominin evolution, including hundreds of species of cichlids that have evolved since the last ice age. And then I also want to just highlight how a couple of other similarities, convergent evolution of body types across depths and other habitats, and then also some interesting, both groups are really notable for kind of unusual reproductive strategies, but in different ways, where cichlids have just incredibly diverse reproductive strategies, often with a high degree of parental care. Sebastes has its own sort of form of that in terms of parental investment in the form of internal fertilization and viviparity. And both definitely have complex mate choice. I think we know a lot less about what that looks like in rockfish than we do in cichlids, but hopefully that will change over time. Like most ideas, good ideas that I've had in my life, my colleague Giacomo Bernardi thought of it 20 years ago and wrote about it in this original short paper in Journal of Molecular Evolution. He and his colleague used mitochondrial DNA and addressed the issue of rockfish species that are differentiated in some way but appear to continue to share genes. In this case, they were writing about black and yellow and gopher rockfishes. But they noted a lot of similarities with cichlids in this way, in particular with regard to separation by depth and also the potential role of sexual selection. So they note that extensive analysis of this is the last line of the paper, extensive analysis of closely related Sebaceous species using fast-evolving markers such as microsatellites should determine if recent speciation events are a common feature of the Sebaceous species flock. So this was 20 years ago, so the microsatellites we've thankfully moved on from a bit. But nevertheless, we have some ways to work towards this goal that they put out 20 years ago. There remain a lot of unanswered questions, including how much current speciation is ongoing in both cichlids and Sebaceous. By the way, for those that are kind of eyeballing that fish there in the picture and wondering why it looks a little uncanny, that's actually not a rockfish or a cichlid. It's an AI mashup that my 11-year-old made in Midjourney. And he also made one showing a cichlid and a rockfish hanging out at Burning Man, which if we have time at the end, I'll show you that one too. All right, so thinking back to our list of big questions in speciation, how can we relate these to diversification in cichlids and rockfishes particularly? And put another way, why do these clades have so many species? So the extraordinary diversity of these groups has generated a lot of speculation that there may be some genomic features that underlie their propensity to diversify. So we're going to look at that in just a second. We have now coming up on enough genomic information to start to answer those questions. Likewise, there might be particular environmental features that have acted to drive allopatric speciation in both groups. In particular, the rise and fall of either sea level or lake level in both clades has been kind of featured in understanding, in particular, the sort of deeper generation of alleles over deeper phylogenetic time. And adaptation to different depths. So depth-separated ecomorphs, like I mentioned, abound in both cichlids and rockfishes. Certain patterns appear to be consistent between them. For example, positive selection on genes in the opsin family. So this was a paper from Arjun Sivasunder, who was in the Palumbi lab when I was there. And in this paper, they showed that rhodopsin genes were subject to adaptive changes associated with depth in 32 species of rockfishes. And then, like I mentioned, both cichlids and rockfishes show some notable features in sexual selection and mate choice, mating behavior, and parental investment. So that's something that we don't necessarily know very much about in rockfish at the moment, but I think it's a really interesting area for research. Okay, and then finally, of course, there's a slew of other kind of grab bag of potential drivers of both pre- and post-mating isolation that we know even less about, particularly in rockfish. In some cases, we're learning more about these mechanisms through examining genomes and characterizing the genetic architecture of back crosses, trying to understand post-mating isolation, for example, by examining recombination landscapes. But getting a handle on all these factors in both clades, of course, is several lifetimes worth of work for many, many people. But luckily, there's a tremendous amount of excellent work already ongoing by research labs around the world that we can draw on to help understand what are some of these features that the clades really have in common. And a common theme here is that cichlid genomic research is quite a bit farther along than rockfish genomic research, but there's still time to catch up. So first of all, we'll take a look at some of these intrinsic features. And this is basically, I took as much literature as I could and tried to digest it and kind of synthesize it into a simple table. So I'm sure I've missed some things, but I would love any kind of comments and thoughts as you look at this. So these are a few of the major features of genomes that we might expect to be correlated with high rates of diversification, and we can ask how they compare across cichlids and rockfishes. Here first are estimates of mutation rates from whole genome sequencing of both cichlids and rockfishes. In the case of the cichlids, they're using Lake Malawi cichlids to better understand between generation mutation rates. And then the Sebastius data is estimated across 88 genomes, so there's probably quite a lot more error around that. But in both cases, these are among the lowest mutation rates reported for any vertebrate. These are extraordinarily low rates of mutation. So it's kind of a surprise. It came as a surprise in cichlids, and then looking at the rockfish, they're perhaps even slower, possibly, than the cichlids in terms of their very low mutation rate. And then same thing for heterozygosity. So if we look at a basic measure of genetic diversity, so here we're comparing population, so it's population level genetic diversity, but compared across many species of cichlids and rockfishes. And similarly, we see that in both cichlids and rockfishes, the average heterozygosity is actually quite a bit lower than the vertebrate average. So of note, that is also true for some other adaptive radiations, like Darwin's finches, it turns out. It's not unusual for species in adaptive radiations to have this relatively low heterozygosity. So overall, that suggests that, first of all, genome-wide heterozygosity, nucleotide diversity, is not driving speciation, right? This is probably a consequence rather than anything causal. And it also suggests those relatively low levels of heterozygosity are not necessarily impeding or limiting rapid adaptation and speciation. We get into a bit more complex features like gene duplications. So the genomes of some of the cichlid lineages that have explosively diversified in Africa and Lake Victoria, Malawi, and Tanganyika, they show an increased rate of gene duplications compared with Nile tilapia and other teleosts. And about a fifth of those duplicated genes show evidence of neo-functionalization. So they're gaining new functions after they're duplicated. And that feature of gene duplication is much less well characterized in rockfishes. But a recent paper showed an association between gene copy number and lifespan across species of rockfish. And the fraction of the genome that's composed of repeat elements is basically the same in rockfish and in cichlids. Regulatory elements for cichlid genomes, they are characterized, at least in those hyperdiverse clades of cichlids, by a greater dynamic in gene regulatory processes than has been observed in other species. So we see increased rates of regulatory element evolution and functionally diverse microRNAs, for example. This is, to my knowledge, has not been synthetically studied in rockfish. So this is an area I think that would be really, really interesting to look at. And same thing for TEs, for transposable elements, for jumping genes and elements of the genome that are moving around the genome. They provide another source of genetic variation. And there's evidence from cichlid genomes that the insertion of TEs near genes has changed expression levels in some key genes. For example, color genes that are involved in species differentiating. And again, in Sebastius, to my knowledge, to the best that I could tell from the literature, this is not something that has been examined yet. The time is right. So we have this somewhat paradoxical finding then that mutation rate and overall heterozygosity within populations are quite low for vertebrates for the species in both these clades. And the question is, how can we reconcile this with adaptive potential, right? Adaptive potential in both these groups. So one other key source of variation could be hybridization between lineages. And this is what I'll talk about for the next little bit, and in particular thinking of sort of bringing it back to some of the research we've done in our group. The role of gene flow and speciation, as I hinted at earlier, is rather complicated. So historically, gene flow has been seen as a process that hampers or impedes speciation, but in cichlids and other taxa, gene flow is now being recognized as a significant factor that helps to establish and sustain genetic diversity across taxa. And genome sequencing across cichlids has shown that they basically all do it. I don't know of a study that has looked for hybridization and introgression in cichlids that did not find it. There's just an enormous amount of hybridization that occurs between cichlid lineages, and a particularly remarkable feature is that they're able to hybridize over relatively large evolutionary distances. And that might be facilitated by the fact that cichlid genomes are also very stable and syntentic. So if you compare, if you're aligning any two cichlid genomes, even quite diverged ones, you'll find that chromosome numbers are similar, genes are syntentic, so they're relatively stable within the group. It's turned out that you can produce viable F2 hybrids between species from Lake Victoria and Lake Malawi radiations. So just to put that into context, that's roughly the evolutionary distance between humans and gibbons. So just really extraordinary degree that they're able to hybridize with each other. And another feature of many of these studies is that the rapid diversification that we see in these groups may be actually sped up by hybridization. So, for example, in Lake Victoria, the diversification pulse was preceded by a hybridization event that included two different riverine lineages. So cichlids from two different rivers, the Nile and Congo, hybridized with sort of the progenitors of the Lake Victoria radiation. And that gave rise to a hybrid swarm where the fixed differences between the parental lineages recombined, generating all these novel recombinations of alleles in the emerging species. And this is illustrated, the way that this is accelerated adaptive evolution is illustrated by this figure here from a paper from Joanna Meyer's group. So this shows long wave sensitive opsin haplotype classes in the Lake Victoria superflock. And what this shows is that these alleles that are essentially helping fish in Victoria adapt to different depths in the water column were derived from hybridization from and introgression from river lineages. So with that, I've been particularly interested in this potential for ancient hybridization to fuel adaptive evolution. In the case of cichlids, in particular, rivers as sort of reservoirs for genetic diversity. And so, for a long time, rivers were ignored in the world of cichlids because the lakes are so dramatic. But that's unfortunate because they present really interesting natural experiments and help us understand the larger context for cichlid radiations. So to study all these questions, I've been working with fishes in the lower Congo River for the last 7 years or so. And here we'll just take a few minutes to give you an overview of some of this work. So this is an extraordinary ecosystem. It's got extraordinary high rates of diversity and endemism, that is species that are found here and nowhere else. It's one of the most dynamic places on Earth. The Congo Basin is roughly the size of the Indian subcontinent, and the entire river basin, all that water, drains through this pretty narrow bedrock-constrained chute, creating some of the most intense rapids and eddies and waterfalls on Earth. So this is just showing you kind of the stretch of the lower Congo here and the topology that it follows. I have a little video that hopefully will play. This is my only video that I took the chance on in the show, but this shows you kind of the scale of some of these rapids. This is right below Pool Malebo, one of our sampling sites. And in a second, you'll see my colleague Ian Harrison for scale. There he is. I don't know if you can see Ian. It's right in the left there. Sometimes it's real hard to get a sense of the scale of rapids if you don't have a landmark. So having set that stage and showed you that very intense environment, giving you an idea of some of the questions that we can ask in this system, I'll just mention a few case studies. So the first is on a genus of endemic cichlids that we worked on in the genus Teleogramma. And this genus is the most rapids adapted of any genus of cichlids that we know of. So it's a really cool little mini radiation of cichlids. These guys are super dorsoventrally compressed. They have these super flat heads, elongate, flattened bodies, so they can essentially just hide under rocks with the current rushing over them. Their bizarre shape caused them to originally be placed in the marine family Labridae rather than Cichlidae. So they're really kind of weird looking for cichlids. But we haven't really understood much about their evolution until recently, because we didn't really have any genomic data. We conducted a RAD-seq analysis a couple years back that gave us a lot of genome-wide data. And what we found was, as you can see here, this is the phylogeny that we came up with for a couple of different known lineages, and then this resulted in the identification of a bunch of new lineages, one of which we've described already, and two of which are in the process of description. But what I want to show you here, too, is these arrows here, which indicate pulses of introgression between lineages. So this is an example here of a radiation that was fueled by both this sort of ancient reservoir of allelic variation and also pulses of hybridization between lineages. When we did a little demographic modeling, we found that the process of diversification in this clade was likely multiple waves of colonization, at least two, maybe more, followed by introgression between lineages that had arrived from different places. Okay, so another little case study here. This is another major piece of this ongoing work to understand cichlid diversification in rivers. Involves a tribe called Lamprologini. So these are the Lamprologine cichlids that in many ways typify what's so incredible about cichlids. They are found in so many shapes, sizes, and colors. They're really kind of the quintessential cichlids in this way. They're ecologically and behaviorally super diverse. They have, some spawn only in shells. Some have sex role reverse polyandry. Some form monogamous pairs. Some have family clans where the elder offspring are helpers in brood care. So just really an incredible gamut of behaviors. So the problem is that like so many cichlids, they're taxonomically an utter nightmare. You'll have noticed in these photos here, so got all of our diversity of Lamprologines, and then we got a couple non-fish on the side here. Well, I mean, only in the sense that all of us are fish. But here, these are my dear, wonderful collaborators on this project. Melanie Stiasny, who leads the ichthyology team at the American Museum of Natural History. Fernando Alda at University of Tennessee, who's really driving this work. And then Noah Kurata, who's a former PhD student in my lab. And then Prasanta Chakrabarty, a longtime collaborator at LSU. So for her thesis, Naoko, which she defended in December, generated 3 RAD data, so that's sort of similar to DD-RAD, to test the hypothesis that high-energy rapids are serving as physical barriers to gene flow that generate genetic divergence in this genus. And so we used that data to assemble this phylogeny, but in the process, Naoko also found that there actually was quite a lot of gene flow between these species. So the species correspond to areas that are separated by rapids. Nevertheless, we see an enormous amount of gene flow still occurring between them. So like I said, very typical for cichlids. Nevertheless, it surprised us that in this context, we see gene flow occurring both downstream and upstream. And how that happens, we do not know. That is like sort of a head scratcher for us, but clearly relates to Paleolithic conditions that might have been different, where the rapids may have been in a slightly different form, allowing multi-directional gene flow. To get even broader, we wanted to take a range-wide view of how river and lake cichlids have diversified and shared genes. So we collected a new data set spanning an even greater evolutionary distance using ultra-conserved elements. And Fernando Alda, our collaborator here, has really been key in collecting those UCE data. Our tree of all the river Lamprologines compared with the lake Lamprologines show that the river Lamprologines are actually nested within the lake, which is interesting and a pattern that we don't see in other parts of Africa. The problem is that we had really low boot support for many of the relationships. So that's often a hint that it's not really the data that are the problem here. We've got quite a lot of genome-wide data, but rather what the data are telling us is that our taxa are not fitting nice and neatly into our typical evolutionary model of bifurcating trees. So we need to use different tools to examine what's going on. And to do that, we can use tools like this one. This is a splits tree network analysis that helps us visualize some of that ancient allelic variation that's shared across lineages And here what you can see is that these squares here essentially depict allele sharing. So larger boxes, like in here, are indicative of more allele sharing at the base of the lineages. So here you can see, for example, a huge amount of shared variation between Lamprologus lethops, which is one of the oldest lineages in the river. And this is one that we've also written about because it's the only eyeless cichlid. It has no visible eyes and no pigment. So really, really adapted to very specific deep conditions in the river. And it's a representative lake species, Neolamprologus burchardi. So these kind of tools can give us some indications of where hybridization and integration are taking place. And we can also turn to the tools of population genetics, right? So this is where we have to kind of leave the classic phylogenetic tools behind and turn to population genetics to help us think about how alleles are shared across clades. So this is an admixture plot that was built in SNMF. Each bar represents an individual sample, and colors represent the likelihood of membership in each cluster. So here we've chosen 10, which was the number of good monophyletic lineages in our tree. So normally you'd expect to see solid color blocks across defined species. What we can see here is a tremendous amount of shared ancestry across just about every species. Again, really matching that split tree that we just looked at, showing how much allele sharing and kind of incomplete lineage sorting is occurring at the base of that tree. And then another useful tool that kind of brings together the phylogenetic framework with the ability to visualize shared ancestry, and we saw this with teleogram as well, is a cladogram. So here we're kind of looking at what individuals or genes are telling us across many, many, many genes, and that gives us sort of a picture of the fuzziness of different relationships. It allows us to see places where alleles have sorted cleanly into lineages and then places where they have not. So places like this, right, up here in this part of the tree where we see kind of this cloudiness suggests that there's a lot of ancestral variation that is still not sorted out into lineages. So it's clear from the Lamprologine analyses that they've hybridized over large evolutionary distances. And the next step that we're trying to work on is to try to understand, in the case of these Lamprologines, how those ancient hybridization events might have, again, fueled adaptive evolution, including in genes related to adaptation to rapids. Okay, so pivoting back to rockfish then, there's lots of super interesting patterns around hybridization in cichlids, but what about rockfish? So do any of the same patterns emerge? Again, in the case of rockfishes, we're a bit behind where cichlids are in terms of our knowledge and the kind of data and analytical effort that have gone into understanding hybridization. Most of the effort's been focused on just a kind of a handful of clades. But there are enough papers that show convincing instances of hybridization across both closely related species and then species that are more distantly related to indicate that this is a common pattern in rockfishes as well. So much of the work that's been done in hybridization rockfish is actually focused on Atlantic rockfish species and Northwest Pacific rockfish clades, less on Northeast Pacific, which have primarily been focused around fisheries applications, understandably, and trying to understand how hybridization might affect our management of fish populations. So I'm just going to highlight a few, again, case studies that I think illustrate some of the more interesting aspects of this clade, and then places where some more research is needed. So these are data that show a structure plot, so very similar to the admixture plot that I showed with the cichlids. This is data from Schwenke et al. In 2018 on copper, quillback, and brown rockfishes in the Salish Sea. And what's notable in this case is that, as you can see from this little tree I pulled from Hyde and Vedder, these are not sister species. So this structure plot shows there's a huge amount of introgression that's going on in the Salish Sea between all these species. But these species are fairly well diverged. So 3 million years or so diverged between the groups. So at least in this example, we see a similar pattern to cichlids, where we have this ability to hybridize over quite large evolutionary distances. And again, that might be facilitated by stable genomic architecture across the genus. And then in another clade, vermilion and sunset rockfish, which are sister species, which are, as far as I can tell, just about impossible to tell apart visually. A recent study by Gary Longo and his colleagues found some level of integration between the two, although maybe not as much as you would expect, along with a whole bunch of cases, probably to be expected, of mis-ID. So this is 231 samples across about 10,000 loci. And in the same paper, they also found substructure both within vermilion and within rockfish, some separated by depth, some separated by latitude, and differing levels of introgression between the two species with the different subgroups of the within species. So this is a complex situation, a complex story that definitely needs more unpacking. Certainly suggests there's very dynamic processes that are still driving speciation in this group. With the help of Scott and his students, I had my molecular ecology class look at some canary rockfish, the sister to the sunset and vermilion clade. So we were focusing on the canary rockfish, and Rachel had done the nice sampling across the coast of California up to Washington. Sadly, we found no population structure whatsoever in canary rockfish, not surprising based on what had come before. What we did find, we threw in a couple of of vermilion and sunset from some recent trips near here as well, and found some kind of odd results. So this is 194 loci so these are microhaplotyped at using GT-seq. And you can see in this top PCA here, we find that Penninger all falls together, very tightly clustered, very low genetic diversity in this group. And then we have our sunset and our vermilion. So again, we're using the identifications that came with the specimens. So with these species that are hard to tell apart, that's always kind of a question that in the back of your mind is there's always the potential for mis-ID. But what I want to just show you real quick are these weirdos in here. So these are 3 fish that we got out of this set of 66 that are, as far as I can tell, F1 hybrids. And so I initially assumed, when I looked at them, it's very clear they're F1 hybrids. They're beautifully heterozygous across all the loci. And I thought, well, those are sunset vermilion F1s, and that's been shown at very low levels in other studies. And then when I took a closer look, one of the parents is a canary in all these hybrids. They're only F1s. There's no back crosses in this set. So this is a real interesting first result. I've stared at it every which way, and I can't interpret it another way, but I'm eager to talk with folks that work on these taxa to try to figure out what's going on here. Anyway, so that was an exciting little project for my molecular ecology class, and they will be presenting some of that at their capstone festival next Friday. And then finally, as we consider, wrapping up here, the potential drivers of diversification in rockfishes alongside cichlids, we can also use some recently generated whole genome data from across a number of rockfish species. This is genome data from Calora et al. To look at, again, how ancestral variation is partitioned across lineages and where it's shared at nodes in the phylogenetic tree. And the main message that I want to impart here is that much like the cichlids, there appears to be a lot of ancestral variation that's shared across these clades. We have these really big swaths of standing genetic variation at kind of the root of the diversification pulse. And so this is definitely a case where we need more genomes represented in these analyses to understand how that kind of allele sharing has affected the adaptive potential in rockfishes. Okay, so to wrap up, we can identify a number of shared features between these two hyperdiverse fish clades, and these first observations might also help point the way towards areas for future research. First, both cichlids and rockfishes are characterized by these depth-separated ecomorphs and a number of convergent features around that. So really interesting work that's being done and to be done on convergent evolution at the level of protein-coding genes and regulatory elements related to depth adaptation. Second, the more genomic data we get, the more we recognize the frequency of hybridization in these clades, including sometimes over these relatively deep phylogenetic distances. Third, even though population level diversity is paradoxically very low, we can see that some of that hybridization, as well as incomplete lineage sorting, the retention of ancestral genetic variation, might provide that substrate for diversification pulses. And then, finally, I haven't had much to say about this because it's really not my area of expertise, but I just find it so fascinating how both clades have these really interesting and unique reproductive strategies, including parental investment and life history correlates that might help act as another engine for diversification. So hopefully that'll be an area for research in the future. So I'm really thrilled to be joining the Moss Landing Marine Labs community, particularly given the deep expertise around fisheries and oceanography here, and I'm looking forward to hopefully working with many of you in the years ahead. And special thanks to Scott both for the invitation here and for kindly giving many of my students some opportunities to experience field work. Look how happy Grace is here on the boat, and Cheryl as well. So thanks to both of you for giving these guys the joy of a day at sea. So, obviously there's a tremendous amount of work to fill in here, and I'm really excited to be embarking on this career grant over the next 5 years to kind of dig in deeper on some of these questions and really apply the tools that we have learned in cichlids to understanding what's going on in rockfish and why they are also such a diverse clade. So with that, I just have so many collaborators that are incredible to thank, including my lab at CSUMB and a lot of colleagues in Congo and at the Museum of Natural History, and funding from NSF and sequencing support from the Museum of Natural History as well. And then I promised you that Burning Man photo. Here it is. So cichlid and rockfish partying together at Burning Man. There you go. And with that, happy to take any questions. Questions? Yes, please.
Hi. I'm really interested about those 3 hybrid rockfish that you were finding.
I thought I might get some questions about that.
Yeah. I guess kind of a two-parter. First, what were they ID'd as initially? Mm-hmm. And then, I'm sorry for the lack of genomics knowledge, but how can you be certain that they're F1 hybrids?
So yes, both good questions. So 2 of them were ID'd as sunset and 1 as vermilion. And for what it's worth, the mitochondrial haplotypes that we did separately do match those, but in sunset and vermilion, there's all kinds of weirdness, you probably know, that goes on with those haplotypes, so it doesn't tell us too much. But that was the original ID. And then with those genomic data, it's possible that, so it's very difficult to tell F1s and F2s apart usually. These are perfectly heterozygous, like at every SNP, they're heterozygous. So yeah, either F1 or F2, but they're definitely not long-term back crosses.
Gotcha.
Yeah.
Thank you.
Yeah.
I'll ask a question.
Yeah.
Thinking about rockfish and their life histories, like in comparison to the cichlids, one major difference is kind of the lifespan, right, and the long generation times and the very episodic successful recruitment events. I'm trying to like wrap my head around like how are those traits sort of line up with the ability to very rapidly kind of adapt and radiate.
Yeah, I would love to hear thoughts from folks on that as well, because I also go back and forth about, longevity is such a kind of key feature that people love to discuss with rockfish and such an interesting feature of their evolution. But is it just sort of a coincidental feature? Right? Is it related in some way to their to the large number of species in the clade, or is it not? Like, is it just a coincidence that they also happen to have this interesting? Because we, to my knowledge, we don't see that the span, the range of different longevities in other adaptive radiations. I don't know of another, but that's an interesting question. It would be worth kind of thinking about. Looking particularly at those couple adaptive radiations that are in the Roboski paper where there's a big size differentiation especially. Like a lot of size evolution might be a way to kind of get at that, whether there's other examples of adaptive radiations where you have very big differences in longevity across species. So yeah, that's another thing to look into.
Yeah. I guess I'm also thinking as well about, like, I mean, it seems like the glaciation cycles were important for rockfish.
Yeah.
And for the cichlids, wasn't there a process of like the lakes drying
Yes.
and refilling? And so I wonder if
Yep.
there's some similarities there in terms of those major habitat changes.
Absolutely, yeah, and I think for both, there is sort of a very kind of coarse pattern that you can see where there's sort of an initial pulse of diversification, creating kind of subgeneric levels of diversification around those glacial cycles, and then a more recent burst of species events that are related to other ecological factors, or all the things we talked about. But yeah, for sure, in cichlids, that's been a point of interest, is understanding lake levels, connectivity between lakes and rivers. So yeah. Yeah, please.
How are you catching the fish in those rapids?
Oh, yeah. Good question. Yeah, very carefully. The center of the rapids tend to be impassable completely. One year, we did hire an extreme kayaking team, but they weren't catching fish. They were using Doppler to try to get depth profiles. We collect fish from the banks, and so we're getting essentially like what in a marine environment would be like beach cast fish. There's all these sort of pools and eddies that form, and there's one site in particular in the Lower Congo that is pretty incredible. We've found several new species there, and we've named one after it actually. It's called Fingu in the local language, and it essentially is a very large eddy that you go and you find all kinds of refuse that's been washed downstream from Kinshasa and Brazzaville. Apparently medical students go there to find corpses to work on. Like if a body gets dumped in the river, it ends up there. Like anything that's in the river will eventually make its way to this particular eddy. And so it turned out to be, not the nicest place to work, but a really good source of specimens. So yeah, we're mainly working on the margins of the river to try to collect what we can. Yeah, we're missing a lot for sure.
Are there ever times of the year when it kind of calms down? Or are those rapids just pumping?
We were there, and that was the calm time. Yeah. Yeah, we only go in the dry season. The wet season, the roads are impassable, and the river is so full that it's not. Yeah, we can't do much. Yeah.
Sorry.
Yeah, no worries.
Mine is maybe a clarification question. So when I think about speciation, right, the barrier to speciation I would think is heterozygosity, because if you have nice even mixing, then you're not going to get that sort of separation of genotypes, maybe. So in a radiation, I guess, why would one expect the heterozygosity to still be? I kind of thought that maybe the lower heterozygosity was
A consequence.
a sign of a recent radiation.
Yeah, absolutely, that can be the case, right? And it sort of depends how much ongoing gene flow there is and relative to the effective sizes of those populations and the kind of the selective environment they're in, what kind of both population bottlenecks but also selective bottlenecks they're experiencing. So yeah, I agree with you. I mean, I think like it's surprising kind of at first blush, but then when you think about the population genetic level of it, yeah, it makes sense that in an adaptive radiation, if you have species that are undergoing these like very like intense adaptive events, that you could get these real bottlenecks that cause lowered heterozygosity. So yeah.
And you kind of already said two keywords in my next question, which was about effective population sizes. So I don't know when hybridization would occur, but definitely like the fish have a lot of really strange reproductive modes. Yeah, different sex ratios, right? And this is coming from somebody who does study fish. Different sex ratios, and then different sort of selective abilities or fitness abilities. So I mean, are the groups that tend to radiate groups that have maybe a more restricted population size than what it looks like, either through bottlenecks or through changes to their sex ratios, where even if you have a lot of fish, maybe certain ones are more involved in reproduction in their population that could cause that change in heterozygosity.
I don't know that I would say that off like in thinking about it for the first time in that framework. But again, I think this is an interesting question that we have enough examples of adaptive radiations now that are paired with extensive genomic data sets that we could actually start to get at questions like that, which would be really cool, Yeah, I think this is a really exciting time for studying adaptive radiations, just because there are so many groups now, from finches to lizards to fish, that we have these, not to kind of focus in on vertebrates, but that's my purview. Yeah, where we can really dig in on the genomes. So, thank you. Cheryl?
So going back to that nice slide you had of the cichlids and the rockfishes and like different aspects of their genome, has that been done across some of these other groups, like with Darwin's finches and other groups? Like, do you see the same sort of similarities across those other adaptive radiations?
I don't know. I mean, I think, so there's certain things that you could, so like, for example, the intrinsic genomic features that would be easy enough, like we're talking about, to look at across some of those other groups. And then I guess the question is, at what point do you kind of get so far away phylogenetically that the comparisons are not so meaningful anymore? But certainly, I think, again, across vertebrates, like, you could probably put together a pretty interesting meta-analysis on a lot of these features. I mean, I know there's been some great reviews of adaptive radiations as they relate to, for example, islands of speciation in the genome. But I don't know that I've seen a sort of a broader scale. It sounds more like a textbook, actually, than a paper. But yeah, I think it's a really good question.
Questions? Yeah. Sorry, I didn't see you. We're hidden. Do you know if there's been any evidence or even any work done on like rapid speciation of cichlids in other areas where they're pretty specious, like in the Amazon or Central America?
Yeah, there has. So again, the Great Lakes of Africa kind of dwarf everything else just in terms of the sheer number of species. But certainly in some of the isolated volcanic crater lakes in Central America, there's been some well-documented cases of ecological speciation, just not quite nearing the number of species, but you have a lot of the same patterns, right? And a lot of the same kind of repeated trajectories of evolution towards benthic and limnetic forms, and different diversification towards different food sources, things like that. It's just that in the African Great Lakes in Malawi and Victoria and Tanganyika, there's just so many species that they essentially fill every niche, right? So they've sucked up a lot of the oxygen. Yeah. Please.
In these really diverse clades, do we see higher rates of sort of like historical extinction among the species because there's so many, or do they sort of persist at the similar rates among other species that are less diverse?
So with rockfishes, I don't know. With cichlids, a lot of these radiations are so recent that we wouldn't necessarily expect to be seeing those pulses of extinction quite yet. What there are some nice documented cases of are species merging together. So you could consider that an extinction event, I guess, right? So in Victoria, I don't know, maybe it's Malawi, I can't remember which one, a eutrophication event essentially removed the ability of cichlids to distinguish each other, two sister species to distinguish each other by color. So the water essentially got so murky they couldn't tell who was who, I guess. And so they merged back into one species. It's an interesting case. But yeah, I think that radiation in particular is quite recent, and so there's not been kind of time for those extinction events quite yet.
Maybe that explains the Canary It's pretty darn big down there.
Any other questions?